We are hampered by our shortcut in time, especially the academic bureaucracy is feeding us up. In Germany, we have the same problems as Carlos has mentioned for Costa Rica. But we can offer that kind of, let's say, evolutionary thinking, what we are trained as biologists. We could offer, for example, devices, access to devices which are not easily accessible, like scanning electron microscopy, molecular tools, and that kind of stuff. And if we link these things nicely together, that would be what we need. And I'm always advocating, but haven't been successful at funding organizations so far. My idea for this is always to have a kind of tandem grants. I call them tandem grants, like a tandem when two people are on a bicycle. That would be grants which also give money for the non-institutional researcher, for example, for traveling and data acquisition, and gives money to the institutional researcher. It didn't come true so far, but I think this is a thing we can lobby for. That would be a real nice thing that we as institutional researchers can write in non-institutional researchers in our grants, just as an idea. I would like to push that. Very much. I give it further to the next one, the magic cube. Better not. I can't.
Yeah, I think it'd be. Yeah. Just a couple of points about that, maybe to start a discussion. The first point is, for me as an academic scientist, the citizen science, or whatever you name it, was really useful, and that way is I'm like scavenging in this group, slime also appreciation, and if I see really something very interesting, I'm like begging for people to send it to me to barcoding, and so far we will have like a new genus published, and many new species are to be published. Yeah, and this is a really, really good thing and advantage for the because you cannot travel that much to all parts of the world, and you don't have all this time. And the other question that I want to ask from citizen science is the credits that they wish to have for their job. I mean, would they wish to be published as the author of species, or to be thanked in other way, or to be co-author in the research publication? Are they interested in that? This is a kind of confusing to me. What kind of things do people want from that? So who else wants to give?
Nice.
Well, I don't think we want to end up in a situation. It's great that people can send specimens for barcoding, but we don't want to end up in a situation where this is the topic of the paper I mentioned, the invariable likeness of sequence-based identification. We don't want to end up in a situation where that is how we identify species and specimens. So those of you who are doing the great work in phylogeny and taxonomy and barcoding and meta-barcoding or whatever, you don't have time, of course, I understand, because you're driven by being able to, you have to publish. You don't have the time that I have, for instance, in you spending time doing microscopy for everyone sends you specimens and you sit down in the microscope. That's not possible. So where do we find these persons that have the necessary skills and time to have that role? They don't grow on trees, so they need to be developed. You have to train them. Somebody has to train people to get to the point where they can actually serve as specialists in identification work, not necessarily in barcoding.
I think that's pretty tricky though, isn't it? Because like I'm a self-funded researcher, and I don't want to spend my time identifying everybody else's material for a researcher. That said, that's a good way of finding what you've got growing or occurring in your local environment. In my state or Australia, is that I can't get out there to get everything. So yes, it would be great if stuff was funneled to me, I could identify it, get it to the researchers. But I think when you're self-funded, you've got other priorities, your own priorities. I don't know. I guess I'm going to keep talking about myself because that's really what I care about at this stage. But what I need is, being a researcher, is I need to have access to researchers to help me continue to grow in what I'm doing. So like that trickier disciplines issue, and I know Eddie's helped us on the slime mold page with that, is where do I take that now? Well, I don't think I can take it anywhere because it's a complex, I can't study that. So I understand that I've done all that work and I understand what's going on in Australia. It'd be great to be able to pass that over to somebody else now to do the work, but I wouldn't consider myself as the citizen scientist. I'm just a researcher, just not quite equal researcher. But you've got your own priorities as well, and that's where I think it starts to become a little bit tricky. So I think we've got lots of levels. We've got the scientific researcher, we've got the self-funded researchers, self-taught researchers, and then we have citizen scientists. So how we get that group of citizen scientists allied to the projects. So again, maybe this is something that can go through the slime mold journal, where you can advertise, basically, this is the research I'm doing, and I am looking for researchers or citizen scientists in the world. So Irina with her Syria could have said, well, I'm about to start studying in Syria. This is what my project's involved, and I'm looking for specimens around the world to help me out. That's how researchers like myself and citizen scientists can get involved in that project. So maybe we need a more formal way of dispersing our projects and then being able to get some assistance. But I don't want to do all the talking. So who else would like to have a chat? Yes, you can say it.
The work that I've done over the past couple of decades has not been a burden, because I've seen more than 200 species that I wouldn't have been able to study unless I spent the time doing that for other people. I wouldn't have been able to be out there collecting all these species, they're using moist chambers and everything. So that is how I learn. So it's yeah.
16-year-old in our state that's really interested in he said to me, I think I found a match for ultra. And we've never seen that in Western Australia. So because he's out there looking, and then he brings it to me, that's the first time I saw it. So I'm not suggesting that I don't want to have citizen scientists helping me. I just think it might become a burden for me, that's all. Because I like to do my gardening and other things as well. Yes.
I want to support Karina's idea about a kind of announcement or a catalog of scientists working with different topics. This may also help academically affiliated scientists to avoid undesirable competition. For example, the last years we have several cases when different groups were working with nearly the same scientific problem, and this causes a lot of ethical questions. And it would be much easier if people are ready to openly talk about their research plans, just to talk about, to put in some kind of online catalog the topics on which they're the project on which they're working right now. And this also may strongly help citizen scientists to find those who can who want and like and can work with the material of the particular genus or family, for example. Those who are new in our community can easily find, okay, I have interest in Licogala, or I have interest in Trichia, with whom I have to come with this material.
Did you want to talk about it?
Yeah, just only a small remark. I think it's a brilliant idea. We should simply suggest to Carlos Rojas in this case, that we ask around in the professional community, which is small enough, what you are liking to work on. He is working right now on the Lycogalas. We are working right now on the biogeography of the Niviculous species and on Lamproderma. I also have an interest in the Bryophyllus Lamprodermas together with Angela. And we could advertise kind of a short summary what we want to do within the next couple of years, let's say a time horizon of 2-3 years, and that we advertise in a paper on slime molds with the point, if you as non-institutional researchers want to have a specimen identified, molecular treated according to the state of the art, give it to these people working in these projects, and then you will get back the results. Of course, the point is those people which advertise should connect this advertisement with a commitment to disseminate these results freely. That's a very important, also ethical point. I've always been adhering to this point. That is, in my eyes, quite important.
So going back to the comment Irina made about where do the researchers or the citizen scientists get placed as acknowledgement for the work that's involved? Pardon? As what?
As authors in the science.
Authors. But you might have 50 people and they're just they're not just collecting, but they're collecting.
Most of which collected all these things.
So, yes.
Sometimes there appears a problem, like for example, in my study, when I work with moist chambers. So there are people who collect samples and there are people who collect substrate for cultivating. So, and that's absolutely another approach. So for myxocytologists, they're collecting of substrates. So our substrates are not the final destination of our work. Mm-hmm. So it's just the beginning, not the result itself. So for example, person, it's only a day to collect bark, for example, from one tree or several trees or like that, and just to give it to researcher. And after that, I started to cultivate it, to collect species, to identify them, to barcode them, to meta-barcode the substrate. So the time I spent to receive the results is much more than for a person who collected during the day. So it's absolutely a different well, taking part, the parts are not very equivalent. Yeah. So, and that was also the question which I was asked once, whether will you include the person who collects the substrate for your project, for example, in co-authors of an article. And I said, no, because, sorry, but yeah, I just explained it like the way I explained it right now. So because for us, it's not the final result yet. It's not the final object. My final object I will receive in a month after that. So if I will be lucky, if the myxomycetes will appear on the substrate just in general. So the impact for the results here is absolutely not equivalent. But another story when, for example, we asked to send some samples, I mean myxomycetes, for barcoding, for example. So it's absolutely another impact because it really requires scientific knowledge, skills, yes, just to find it, to know where to search for them. You find them in field, you identify it more or less, at least to genera. If you don't even know species, but you can at least say what generates that. So then you just brought it to your laboratory or to your home microscope and look at it there. So you also spend time, then you send it, and the researcher, well, the impact here is more or less equivalent. So that's another point of view. So it's another question. So in this case, of course, yes, you have to discuss it and just to decide which author you, as first author, who is the first author, who is the second, who is the mentor, and that's all. So yeah, so here's the impact. It depends on the impact of the study you take.
So your first case study that you gave us there, you would put those people that collected the substrate over 1 day, etc. Would you acknowledge them?
So yes, I told in my case, I was telling them my theory about my vision of the situation. And after that, I mentioned, of course, I will mention those people in methods, for example, who collected, where collected, and yes. So in methods, so at least in methods and in acknowledgements. So, but there's also part that if, for example, people collect the samples in nature preserved territories, for example, in national parks, for example, or regional parks, something like that, where there's also a kind of scientific component of, well, just research, well, also researchers, but for example, in another field of research, for example. They will be interested to receive the list of species when the work will be finished. So this would be also very interesting for them to receive the list of species when the experiment will be finished, and I will have a list of them. So that's, I also mention all the time that if you are interested, the paper will be where the data will be mentioned. So, and after that, I will give you the list of species which I found. Of course, I will not hide them, so they will also have the some material interesting things to help the researchers.
Sounds like you've got it well sorted. So there's Mark and up here.
Yeah, it's absolutely true. We cannot include everybody as author. I was a little bit too quick to tell that. For many studies, we have constraints, and in academic research, usually a lot of research is done in the frame of PhD thesis works. In our university, at least, and many universities are similar in that a PhD candidate is required to have at least 2 papers with himself or herself as a first author. And we also have a provision. I served often in thesis committees and saw that if the list of authors is longer than 10 people, there are nasty questions. Oh, what is your own contribution to a multi-author paper? What we should always do is acknowledging the people, even if they are only in parentheses developed, or in quotation marks developed or contributed substrates. But some journals allow something in between with contributions of. You have 5 authors, and then comes with contribution of, and you have another 10 authors, but it is rather rare. So when we do such a proposal, we also have to agree on a point. What do you need to become a co-author? I would say if you, for example, contribute more than, I pulled a number now out of the air, but something like that, if you contribute more than 10% of the data of the study, you should be a co-author, something like that. There has to be a kind of threshold to be set. And the main point is, last thing, to communicate in advance. That's it.
Just a remark. And if your collection is a new taxon, then you are co-author of the taxon, but not necessarily of the paper.
Yes, thanks. Good point.
Send it up here and then I'll bring it down.
So I think that it is not the question who is the contributor. So if he's the citizen science or academic researcher, but what is the input to the given, to the publication, because we also share collections between academic researchers, and not all of those who gave somebody collection is a co-author, sometimes only a knowledge. So it means, I think, it depends on the input to the particular study. And also, I think it has both sides.
the authorship is not only an honor, but also a responsibility. So I think that not all people want to be responsible for the results they don't fully understand. So sometimes they prefer to be a knowledge only because they don't want to give their own responsibility for conclusions and so on. So I think that we cannot create automatic rules. It just every single case is different.
Yeah, and the Vancouver Convention provides some pretty specific recommendations for criteria necessary to become an author or a paper as a person. And you have to contribute somehow intellectually. And just if you happen to collect something and gave it to someone and it ends up being interesting, that's not qualifying for authorship.
Yeah, so true.
I'm not a mixomethodologist, but I'm also a taxonomist of Basidial fungi working here in Tartu University. And I would like to chip in maybe a bit anarchic thing. So I would like to remind you that when we are talking about scientists and about citizen scientists, we need to remember that co-authorship and citations, they are currency to us. And our academic world is kind of weird world, and the situation of currency is also very weird. Yes, and we are bound to be weird, but we are not bound to impose this weirdness on people who have nothing to do with that, right? So we have rules when we include people in co-authorship in this case, when we exclude from co-authorship in that case, etc., etc. But these people usually, if they are citizen scientists, they don't care. So we kind of impose our rules on the world of normal people, right? And I think in this case, we should just not think about these rules and include as many people as possible, because everyone understands that the main work on the paper is done by the 2-3 people, and the last was given money, right? No one will ask why you have 30 people, why you have 50 people, because these papers are very common. We have plenty of papers with these numbers of people, and still we understand how many of them do the actual work, and we actually can specify who contributed to the paper equally, right? So I think in this case we should be less weird to people and just give them co-authorship. Thank you.
So just for my own edification, me being a armchair amateur citizen scientist, whatever you want, what would be the benchmark for contributions to then be listed, either as a co-author, a acknowledgement, or a contributor?
Who would like to answer that question? Right, well, in the case of the only 2 species I've described, I found it. I did probably 80% of the work, and then my collaborators helped me with the bits I couldn't do. So I would not be able to publish without them. So for me, I know that they're I know they're scientists, and I know they're very well known. And so it's probably easier to put a scientist like that on your paper. But even if it was a citizen scientist who was helping me to write that paper, and even if it's to the point where I'm using them as a sounding board, helping me write up, etc., etc., I would put them as a co-author. But I also have people that help in other ways, and they don't have an expectation to be a co-author. They're very happy to be acknowledged. So anyone that helps me in the paper, the last paper, I actually commented that I was really lucky because my husband drives me around the countryside collecting substrate. He's actually really important to my research, but he's not a co-author. He's always acknowledged. So it's a matter of finding the right spot. Now, it may depend on the researcher that you're dealing with. And I mean, we've heard a number of different views here now as to where they would place you, or you might want to place them. For the scientists that helped me, I approached them and said, would you help me co-author this paper? And look, they're normal people like the rest of us, so we can just ring them up or email and say, do you want to help me out with this? And usually they're pretty good. Yep.
I appreciate your response. I can now see and understand that's not binary. But one other question or comment, I guess, towards you, Edmund. You say part of the problem is that you're the one who's more or less responsible for most of Norway for helping people identify to species. Me being pretty new to the whole world, I've had to learn a lot myself. So are there any good resources to understand what to look for microscopically and examples of that, which I found personally kind of hard to find online to better my studies and make me a better identifier?
The question was resources or how to learn how to do microscopy and morphological studies on mixed mycetes.
Yes.
Well, it's difficult to answer because it's a question of rehearsal and also experience. There are different structures to be focused on for different groups of mixed mycetes, and new methods like polarized light, Dmitro, is turned out to be useful for Leucogala. We never used that before, at least I didn't. So I have had to play around with polarization filters on my microscope in order to study the Leucogala. And people in Norway, they take microscopy courses, general courses, even for fungi. You're using different dyes and different methods, some with phase contrast, some not. And for ascomycetes and or basidiomycetes, there are different methods and different structures, and even in various genera, also in mixed mycetes. And you don't learn how to do microscopic studies on mixed mycetes in a 2-day course. When it comes to online resources, I don't have a clear answer for you, unfortunately. Just otherwise, in general microscopy, you need to learn how to use your microscope and adjust it correctly, because a lot of people that I see doing microscopy, their microscope is crap. They just turn buttons like haphazardly, and I don't understand how they can see anything. So point one, learn how to adjust your microscope, have a decent microscope, and then you have to just learn by doing it. Yeah, practice and ask people who knows how to do it.
Just to pick up on this, I just want to say how we work in Belgium. We have a regional mycological society, and we give lots of courses there. I give over the years, over 10 years already, myxomycetes identification course, and that's also a follow-up of the fungi identification course. So other people give that. We give microscopy courses, and that's all for the members. And the members just pay their annual fee of 23 euros. And they get all that for free. I have also a website where I put in details how to work with, how to identify myxomycetes, how to do moist chambers, and so on. So there's lots out there, I think. But if you have a local mycological society, I would go there and start from there. Maybe they work the same way, but we have always worked that way. We know that the people, the lovers, let's say, of mixos and fungi. We need to care for them. So we really need to facilitate that they can work in the best way possible. So and I'm also now working. I started as an amateur, so and now I'm working as a professional mycologist. So but I always know my roots. So so I know I keep on giving those courses and helping people, and it pays off. You see that they enter their data, they send specimens, they ask questions, they work the right way. Yeah, thank you.
Yes, we have like 35 local regional mushroom societies in Norway, and many of them are now requesting microscope courses, and we keep those courses, but that's on general microscopy and mostly for fungi and basidiomycetes. So I'm not saying that's not important, it is. But when people have gone to these courses and they send me some micro images from their samples of myxomycetes, I can't really see anything because they haven't really learned how to make good slides and, you know, samples and good pictures and looking for the right structure. So that's the next step after learning how to do microscopy in general.
Microscopy courses during this. So, do you teach any mixomethodology during these courses, or it's just general mycology, or what?
One course many years ago. Since then, I haven't given course. Many lectures. Really the same. Mistakes involved.
Well, looking forward, I think we should one day have the mixomethodological foray or whatever you name it in Norway. It would be super fun.
Okay, we've got about 5 more minutes, I guess. Yes.
I'd like to take a role of advocate diabolis, and if we name people who collect the data, scientists, but are they scientists? Yes, they can collect data, identify some species, but in my opinion, difference between scientists and amateurs is that way that amateur can discover some fact, but this fact this fact don't be link with another fact in some systems. And scientists link these facts in a system and make some conclusions. So is a citizen scientist scientist?
I would say that people who just collect something and without any knowledge say, okay, Does anybody know what is it? It is not yet a scientific work, of course, but when people collect a lot of material, able to identify some strange things and ask the expert about this is already a first step in scientific work.
I would agree.
I wanted to say something similar to Dimitro because I have some very, very nice amateur friends, I can't call them, and it's impressive. So they can see things that I cannot see sometimes. And my work is as a taxonomist. So I am supposedly the scientist, let's say, and they have found things that it's like, wow, how did you see this? So sometimes they are more than us.
Academic scientists, you have to do a lot of work, like in the lab, the writing, whatever. And sometimes the amateurs have a luxury to go just day after day and collect something on like 1 square kilometers, and they know literally more than science because they see the plasmodia, the fruiting bodies, maturing fruiting bodies, etc., and they are really experienced in that. You, like academic scientists, just doesn't have the time to gain this specific knowledge. So yes, that is just a different type of science, I would say, or expertise.
So what we're saying is it's a bit like citizen scientists, curiosity-driven researchers such as myself. You can't pigeonhole anybody in any particular grouping. And really then I guess it's up to the scientist to negotiate that with the citizen scientist at the time of the beginning commencement of the project. So yeah, I think that the point here is that we can't pigeonhole these citizen scientists into whether they're using their brains or not to assist you in your endeavours. So I don't know what we might have achieved from this. I guess I might ask Irina. I'm going to put her on the spot. Sorry, you're looking at the phone. So in your project, you've already mentioned people sending you material for your PhD work. So did you advertise your PhD to the community and then ask at the time, so I'm talking about the science community and the citizen science community, do you think it would have been really helpful if you'd done that right from the beginning to be able to communicate that and then be able to get citizen scientists and or other, doesn't matter what researcher, citizen scientists, researchers, call them what you like. If you'd had that occur right at the beginning of your project, that would have been really helpful for your project. Yeah. That's right. Yep. Okay. So I think that's probably a good way forward for researchers to be able to advertise right from the beginning what they want to study, what they want to achieve, who can help them, and also at the time they can negotiate, authorship at the time. But that could also change, of course. And then there's on the other side is researchers, curiosity-driven researchers, such as myself, being able to then understand who's studying this material, these species, etc., or genera, whatever, and to be able to get in really early to assist as well, and vice versa. If I, find a particular new taxon that I know who to contact. So I think we need to wrap it up now at 6 o'clock. I think that's been a really interesting conversation. I think we did achieve one or two things out of that conversation. And we'll just have to figure out how to go forward from there. And I was just going to say, Ilan, is just put more questions onto the Slime Mold Facebook page, perhaps to get a little bit more assistance. Because there are people out there that will be able to help you. I mean, I know you already do, but when it comes to microscopy, even if you put something up and say, I can see what you all can see. What do I need to do to make this? What can I do? that sort of thing. So